Introduction to Microbiology. This lecture mainly on medical bacteriology. So what's microbiology? Microbiology is the study of microorganisms. So most pathogenic microorganisms are in cellular form or acellular form. The cellular organisms which causes pathogenesis are pathogenic to humans or bacteria, parasites and fungi, whereas acellular particles which causes infection in humans are virus and prions. So the bacteria are very tiny organism which is not visible to our normal eye. So the bacterial size is 1 micrometer in size, which is similar to the eukaryotic mitochondria size. And the smaller than bacteria are viruses and later on protein, lipids and atoms. First type of cells on earth are prokaryotes. The life on earth started 4 billion years ago, and the first life on earth are prokaryotic type of life. So up to 2 billion years since the starting of life, we have only prokaryotes. Then thereon it shifted to eukaryotic type of cells. To classify the cells on earth in 1980, Woos defined 3 cellular domains. One is a bacteria, other one is archaea, and the third one is eukarya or eukaryotes. So the 3 domains, archaea, bacteria, both are prokaryotes, and the third domain is eukaryotes. In eukaryotes we have protists. The examples for protists are algae, protozoa, fungi. A lot of fungi are good for humans and very few fungi are pathogenic to humans. Plants, animals. So 3 domains are archaea, bacteria, eukaryotes. Archaea. Archaea is different from bacteria, which is a prokaryotic in nature but different from bacteria. It lives mostly in harsh environments, and nowadays they identified that it is also living in normal environments where the eukaryotic cells exist. So there is no archaeal pathogens or parasitic in nature, and these archaeas are methanogens. That means they produce methane gas, so which is useful for biogas and treating the sewage. So the bacteria, the archaea, lives in very harsh environments like hot springs or acid mine drainages, very low pH areas, and this is an archaeal organism which lives in human gut, but it is not pathogenic. It helps in digestion of food inside the human gut. This archaeal organism is Methanobrevibacter smithii. So bacteria. Bacteria are prokaryotic type of cells. So what is prokaryotes? Prokaryotes are the cells with no nuclear membrane and no cellular organelles. And the bacteria lacks sterol in their cell membrane, whereas humans will have cholesterol in their cell membrane. The bacteria won't have any sterols in the cell membrane. They have a circular type of DNA, whereas the humans have linear type of DNA. The bacteria has ribosomes, which is 70S in nature, whereas humans 80S in nature. The bacterial normal size is 1 micrometers in size or 1 micron in size. So the difference between archaea and bacteria. So this archaea and bacteria, they differ in peptidoglycan structure wise. The peptidoglycan of bacteria or cell wall of bacteria is made with muramic acid, which is murine in nature. That means tough, murine in nature. Whereas archaebacteria, they can have pseudomurine in nature, not exactly like peptidoglycan of bacteria, no muramic acid. And the bacteria and archaea differs with RNA sequences, 16 years RNA sequence even. So that's why they are present in 2 domains. So the major differences between 3 domains, eukaryotes, bacteria and archaea, are cellular configuration. The eukaryotes have eukaryotic type of cells, Whereas bacteria and archaea, they have prokaryotic type of cells where there is no nuclear membrane and the nucleic acid is spread all over the cytoplasm and no cellular organelles. Whereas eukaryotic, they will have a well-defined nucleus with nuclear membrane and membrane bounded organelles. Nuclear membrane present, absent for bacteria and archaea. Chromosome nature linear, whereas bacteria and archaea, they are circular. Cell membrane consists of sterols, whereas bacteria and archaea, they won't have sterols in their cell membrane. The ribosomes are 80S in nature, whereas bacteria and archaea are 70S in nature. Whereas eukaryotes will have cellular organelles like mitochondria, they will have endoplasmic reticulum, rough Golgi apparatus are present, some membrane bounded organelles, whereas bacteria and archaea, the membrane bounded organelles are absent. And the amino acid initiating protein synthesis is through methionine in eukaryotes, whereas in bacteria it is with the N-formyl methionine, whereas archaea it is same as the eukaryotes methionine. So to classify bacteria, there are 24 phylums of bacteria. To classify these bacteria, in 2001 a scientist called Burgess, he organized the taxonomical classification for bacteria, which we used to call it as Burgess Manual of Systemic Bacteriology. And according to this classification, for example, if you take Escherichia coli, E. coli, which is the most common urinary tract infecting organism in females. So if you want to classify the E. coli, so the domain of E. coli is bacteria, so it comes under bacterial domain, that is prokaryotic type of cells. Coming to the phylum, it is Proteobacteria. Coming to the class, it is Gamma Proteobacteriae. Order Enterobacteriales. Family Enterobacteriaceae. Genus Escherichia, and the species is E. coli, Escherichia coli. So coming to history, in 1665 Robert Hooke first observed the cells in plants. So he used to call it as little boxes, and later on it is defined as cells. And he observed all these plant cells with 20x magnification. And he used very small simple magnifying glasses to observe these cells. And he is the first person who suggested that all living things are made of cells. In 1665, Robert Hooke was the first person to observe the cells. And it is the microscope used by Robert Hooke in 1665, and he wrote a book, Micrographia. So by reading this Micrographia, there is one person who got inspired to observe the tiny organisms, tiny parts, tiny structures. That person is Anthony von Leeuwenhoek, who is a Dutch scientist, and he got inspiration by reading this book, which is written by Robert Hooke, R. Hooke. So he is the Anthony von Leeuwenhoek. So he first started using this microscopes, tiny laser microscope, where this is the lens area, and he used to keep this specimen here, and he used to observe through this lens. And he observed bacteria, protozoa, sperm cells, blood cells, microscopic worms. This is the book that was written by Anthony von Leeuwenhoek. He is a Dutch scientist, works in a glass factory. So the next person who developed microbiology, so he is Louis Pasteur. He is the father of microbiology. He is the first person who proposed the germ theory. So the germ theory is nothing but any infection or disease is caused by some kind of microorganism that is called as germ. He first proposed it, and later on it was approved by his associate Robert Koch. So Louis Pasteur explained about fermentation. He explained about the pasteurization to prevent the spoilage of wine, milk, and he developed a vaccine for rabies. Robert Koch. So Robert Koch is the first person who proved the germ theory, which was proposed by Louis Pasteur, and he created postulates. So next slide we are going to discuss about the postulates of Robert Koch. So Robert Koch identified anthrax, tubercular organisms, cholera organisms. He is the first person who developed microbiological media, the media where we can grow the bacteria outside of the host. He also developed stick plates for pure culture, for extracting the exact organism, the pure organism. He develops the stick plate type of culture. Koch postulates. There are four Koch postulates which was defined by Robert Koch. So according to it, the specific microorganisms is present in all cases of the disease. That means whenever there is a lesion, in that lesion, the lesion causing organism has to be present in that lesion. And according to the second postulate, the organism can be obtained in pure culture outside of the host. So whatever the organism that is present in that lesion, it need to be extracted, isolated, and it need to be cultured outside of the host as a pure culture. So third point, organism when re-inoculated into a host causes the same symptoms. So the cultured organism, the organism which is grown on the culture media, whenever it is inoculated in an experimental animal, so it needs to produce the same type of symptoms and same type of lesions in that experimental animal. And the fourth postulate is organism can be isolated in pure culture from experimental infected host. So the organism which causes the symptoms in experimental animal, it has to be isolated and it need to grow in the outside culture media again. So according to this four postulates, he proved the germ theory, that germs causes disease. So for every bacteria, there are few things that are required for their replication, for their survival. One is a nucleoid material, that is a chromosome DNA material, and it must have a ribosomes, it must have a cell membrane, it must have a cell wall, and other surface layers like molecules like capsule or slime layer need to be present to grow and to replicate inside the host. So bacterial structure. So what are the components of bacteria? What are the parts of bacteria? So bacteria will have a cell membrane. It will have a cell wall. All bacteria will have a cell wall. We could call it as a peptidoglycan layer. And on top of this cell wall, some bacteria will have a layer called capsule, or some bacteria will have a slime layer, capsule or slime layer. If it is thick, we can call it as a capsule. If it is thin, we'll call it as a slime layer. And some bacteria will have flagella, one or more flagellas. Some bacteria will have pili, hair-like projections, which starts from the cell wall, and we can call this pili as fimbria even. Fimbria or pili, both are interchangeable terms. And bacteria will have a mesosomes. Bacteria will have inclusion bodies where they will store a lot of nutrient material. Bacteria can have ribosomes which helps in protein synthesis, and some bacteria will have extra nuclear material, extra DNA, extra bit of DNA. We'll call them as plasmids, and all the bacteria will have a nucleoid material, a circular DNA material inside them. So altogether, this is the bacterial structure. These are the parts of bacterial structure. Inside the cytoplasm, they will have a lot of vitamins, a lot of nutrient material, a lot of macromolecules, a lot of electrolytes. They'll have a lot of material and water included. So this is the diagrammatic representation of a bacterial cell. In this cell, you can observe there is a thick capsule above the bacteria, on the surface of the bacteria, and this is the cell envelope, which includes a cell wall, a periplasmic space, and a cell membrane. When you zoom this portion, you can observe like this, and inside the cytoplasm, you can have inclusion bodies, lot of inclusion bodies where they will store a lot of material and nutrient materials or waste materials, and they will have a lot of ribosomes, small dot ribosomes, and they will have a nucleoid material, genetic material, which is spread all around. The prokaryotic cells won't have a cell membrane, they'll spread all around the cytoplasm. They'll have mesosomes, which is nothing but imagination of the cell membrane. And on the other side, you can observe a dividing bacteria, replicating bacteria. It is replicating by binary fission with a septum. You can observe the septum, which is dividing these two cells. Inside you can have a genetic material here. On top of it, they'll have a lot of Pili or fimbriae. You can observe this. A lot of ribosomes. The small dots are called as ribosomes. And the cell components. For our easy understanding, we can divide the cell parts of the bacteria into three groups: cell surface appendages, cell envelope, cytoplasm. In cell surface appendages, we'll have flagella. Second one is pili or fimbriae. Whereas cell envelope, you'll have capsule or a slime layer. We'll have a cell wall, which is peptidoglycan layer, and cell membrane. In cytoplasm, we'll have the important organelles are nucleoid material, circular DNAs, ribosomes, which are 70S in nature, plasmids, mesosomes, inclusions, and other macro and micromolecules inside the cytoplasm. So this is the bacterial structure. So the first part of bacteria, that is flagella, which is surface appendage. Flagellas are mainly useful for motility of the organism. They help in motility. They always arises and attached to cell membrane, not to cell wall, cell membrane. They are powered by proton motive force. That means they normally get the energy to push the bacteria by proton motive force, which is a chemoosmotic potential. Normally in eukaryotic cells, the flagellas will be pushed, or flagellar motion will be achieved by ATP synthesis. But here in bacteria, they used to get the energy by proton motive force. Flagellas are antigenic in nature. They can stimulate the human immune system. They're antigenic in nature, and we can observe the flagellas in fluid wet mount media. So you can see the flagellas, bacterial flagellas. So these flagellas are four types: monotrichous, peritrichous, locotrichous, amphitrichous. Monotrichous means, you can observe here, this is the organism, the big structure is the organism, and they will have a single flagella at one end. So we can call it as monotrichous, which has a single flagella at one end. The best example is Vibrio cholera. Peritrichous organisms. Peritrichous means, this third one, you can see the organism which has lot of flagellas on all around the surface. Lot of flagellas. That is peritrichous all around. The best example is E. coli. Locotrichous. The locotrichous is the second one. For locotrichous, you can see abundant flagellas at one polar end. A bunch of flagellas at one polar end. The best example is Helicobacter. The Helicobacter is the one which causes gastric ulcers in humans. The fourth one, Amphitrichous. We can see the bacteria here with flagellas on the two polar ends, not on entire surface, only on polar regions. Amphitrichate. The best example is Rhodospirillum rubrum. So you can watch a short movie of flagellated organisms, how they will move. You can see the flagellas are moving counterclockwise and the organism is pushed, and here it is going with clockwise, counterclockwise here. So here is the flagella movement. We can discuss a little bit about flagella movement. So this is an organism with peritrichate flagellas. All these flagellas are bunched together, and all these flagellas are rotating in counterclockwise direction. Whenever they move counterclockwise direction, the organism will get a forward movement. For counterclockwise direction, the organism will move forward. Here also a monotrichous organism which has a single flagella, and this flagella is also rotating in a counterclockwise. Whenever it rotates with a counterclockwise direction, the organism will move forward. The movement will be towards a forward movement. And here is the third organism, which is a peritrichate organism. But here for this organism, there are a lot of flagellas on the surface, peritrichous in nature. Each flagella has different oriented movement. And this flagella is clockwise, and this flagella can be in different way. This flagella can be in different way. Whenever these flagellas are moving, rotating in different orientation, the organism will get a tumbling movement, shaky movement. And the fourth organism, and it is also monotrichate flagella. If this flagella rotates in a clockwise direction, clockwise rotation, the organism will move towards backside, backward movement. The next component is fimbriae and pili, and we can use these terms interchangeably. They are hair-like structures present on bacteria, and they always arise from cell wall. The main function of this pili or fimbriae is attachment and making colonies, colonization, and they also inhibit phagocytosis. Especially the best example for inhibiting phagocytosis by pili is Neisseria gonococci, which causes gonorrheal infection, Escherichia coli, and strep pyogenes. In strep pyogenes, pili and M protein both combined together causes inhibition of phagocytosis. And these pili are antigenic in nature. They can stimulate the immune system. And there are special pili you can observe in some bacteria like E. coli. They act as sex pili or F pili or X pili or pili. And this sex pili always helps to exchange the nucleic acids from one organism to other organism. This sex pili connects the two different bacterial organism with the help of this pili, and through this pili the nucleic acid will be transmitted to the other organism. So the next component is capsule. In cell envelope, the first component is capsule. These capsule, some bacteria will have the capsule, and these capsules, whenever they are discrete, detectable layer of polysaccharides which are present deposited outside the cell wall, we can call it as a capsule. In simple form, if the polysaccharide material which is present on the cell wall, if it is they're thicker, rigid in nature, we can call it as a capsule. If it is thin in nature, which is thin and matrix, like a thin layer, we can call it as a slime layer. The slime layers most of the time makes biofilm, right? Less discrete polysaccharide or matrix which embers the cell is called as slime layer or a biofilm, which makes biofilm. They are antigenic in nature, inhibits phagocytosis and desiccation. Most of the time the biofilms makes a dental plaques. Whenever the organism which produces slime layer, they can make biofilm. Especially this biofilm, best example is dental plaque. So the organisms which are growing in biofilm are resistant to the environmental factors. They are protected from environment for themselves and other bacteria even. And the capsules can be easily absorbed with the help of Indian ink and Quillen's reaction, but the slime layer can't be visible. The capsulated organism, most of the time, they cause meningitis because they restrict phagocytosis. They can cause meningitis. These organisms can cross placental blood-brain barrier and they can cause infection. And the capsules mainly made up of polysaccharides or hyaluronic acid, D-glutamate. The one organism which is important to make a capsule D-glutamate is Bacillus anthrax organism. So here is the capsulated organisms with Indian ink staining. These are capsulated organisms. This is the capsule, and they are dividing cells, replicating cells, binary fission. Here is one more cell with bacteria with the capsule. Here is the capsule. Here is a big capsule here, and on the other side you can see a big capsule here, a hazy material here. This entire thing is a capsule, hazy material, and this is the cell wall, and this is the septum, dividing, replicating cell, septum, and this is the dental plaque. which is stained with a red dye, which is harmless in nature. The red dye is not, it is harmless. And this is the dental plaque, which is nothing but a biofilm. So organisms which produces slime layer, they can make a plaque or a biofilm where most of the organisms can grow comfortably by resisting the environmental factors. And these are the organisms with slime layer. And here is the Bacillus anthrax colonies, which are mucoid in nature. So the next component of bacteria is cell wall. Cell wall is very rigid in nature. The main purpose of the cell wall for bacteria is to prevent osmotic rupture. There will be a lot of pressure, osmotic pressure from outside on the bacteria. So to prevent this osmotic rupture, they must require a protective layer which is made with cell wall. So the cell wall we'll always call it as murine. Murine is nothing but a unique type of peptidoglycan layer. And this peptidoglycan layer is mainly made up of peptide and glycan. Glycan is made up of disaccharide sugars and which are linked with 4 amino acids, short chain amino acids. We'll call it as peptide. So combined together, peptidoglycan. This peptidoglycan is synthesized with the help of 3 enzymes which are present in periplasm or from the cell membrane. Those 3 enzymes are transglycosylases, transpeptidases, and carboxypeptidases. These 3 enzymes can make peptidoglycan. So there is one pharmacological interpretation here. The beta-lactam drugs always destroys this peptidoglycan layer or inhibits the synthesis of peptidoglycan layer. How they can achieve this inhibition of synthesizing peptidoglycan layer is the beta-lactam drugs always suppress the action of transglycosylases, transpeptidases, and carboxypeptidases. Whenever the action of these 3 enzymes is inhibited and there is no synthesis of peptidoglycan layer. So in that way, the beta-lactam drugs like penicillin inhibits peptidoglycan synthesis. So the peptidoglycan layer is made up of 2 sugars disaccharide, that is N-acetylmuramic glucosamine, that is referred as G, N-acetylmuramic acid, that is referred as N, and these are associated with 4 amino acids, short chain amino acids, which are peptides, that is L-alanine, D-glutamic acid, diaminophilic acid, D-alanine. So these 4 amino acids most of the time they associate with N-acetylmuramic acid, N. So here is the bacterial cell wall explanation. So this is the peptidoglycan, glucosamine, N-acetylmuramic acid associates with 4 amino acids. And here there is one more chain of peptidoglycan, and these 2 peptidoglycan layers are connected with the help of a side chain amino acids. And these side chain amino acids attaches these 2 peptidoglycan layers with the help of transpeptidase enzyme. Here is transpeptidase enzyme, carboxypeptidase bonds the 2 tetrapeptide amino acid chains. So whenever you see the picture, you can see one more diagram here. Glucosamine, muramic acid associates with 4 amino acid which are attached with an amino acid side chains. 4 amino acids associates with 4 amino acids and accessory amino acids. So in this way they are bonded each other and forms a thick peptidoglycan layer which is very rigid. And there is one more molecular representation of peptidoglycan layer. This is muramic acid, glucosamine. Muramic acid is always associates with 4 amino acids here, 4 amino acids, and which are linked with the accessory amino acids. and in this format they mix peptidoglycan layer, a thick peptidoglycan layer or a cell wall. And here you can see one more diagrammatic representation. So the action of beta-lactam drugs and lysosomes. So how this lysosomes breaks the peptidoglycan layer, how the beta-lactam drugs acts on peptidoglycan layer of bacteria. So the penicillin is the best beta-lactam drugs widely used. Whenever the penicillin is given for any bacterial infection, especially gram-positive bacteria, the penicillin drug breaks this accessory amino acid chains which attaches to amino acid tetrapeptide chains. So they will break them. So whenever they are broken, the peptidoglycan layer will be separated. The peptidoglycan chains are separated, weakens. So the bacteria will be osmotically destructed. And whenever the lysosomal action, because of macrophages and neutrophils, whenever they engulf any bacteria, how they can kill is the lysosomal enzymes are secreted and they'll break the bonding between glucosamine and muramic acid, N-acetyl muramic acid. So the peptidoglycan layer is destructed and the bacteria will be destructed. So the cell wall in bacteria is of two types of cell walls. One is gram-positive cell wall, another one is gram-negative cell wall. Gram-positive cell wall, in staining technique, one special staining technique called gram staining technique, it will be observed as blue or purple in nature gram-positive organisms, whereas gram-negative organisms are observed as red in nature. So here you can observe a diagrammatic representation. The gram-positive cell wall, you can have a cell envelope here. This is the cell membrane, and this is the thick peptidoglycan layer, the cell wall for gram-positive bacteria, and here is the capsule. Whereas gram-negative organisms, cell wall, they'll have a cell membrane and they will have a gap, periplasmic space, a gap here, and they have very thin peptidoglycan layer, and they'll have one more extra membrane called outer membrane. This is inner membrane, this can be outer membrane. The cell membrane is inner membrane, and which is there is one more membrane called outer membrane which is present outside, towards outside of the bacterium, and on top of it some bacteria will have a capsule. So we'll learn about this. So how we can differentiate gram-positive and gram-negative organism is depending upon gram staining. So first we need to learn what is gram staining. So in gram staining, the specimen is taken and it need to be made into a small smear on a glass slab. So after making a smear and it need to be added with primary stain, crystal violet, a small crystal violet drops which are violet in color need to be added to that smear. So after adding the crystal violet drops, we need to wash that smear for 1 minute, and after washing that we need to add iodine to this smear. After adding iodine, it need to be kept for 1 minute, and after 1 minute it need to be again rinsed with water. Later on we need to add a bleaching agent or discoloring agent, that is 95% alcohol or acetone or ethanol, for 30 seconds and wash it, and afterwards we need to add the last stain called counter stain, saffron. So after adding saffron, wait for 30 seconds and you can observe the smear under oil immersion microscope. Whenever you observe under the oil immersion microscope, if you see the organisms are purple or blue in color, we can call those organisms as gram-positive organisms, gram-positive. If we see the organisms are pink or red in color, we can call them as gram-negative organisms. So gram-positive organisms are purple in color or blue, gram-negative or red in color under the gram staining smear. So here is a small short animated video of gram staining technique.
The gram stain is the most widely employed staining method in microbiology. It is a differential stain because it divides bacteria into two classes, gram-positive and gram-negative. In the first step of the gram stain procedure, cells from a fresh culture are transferred to a clean slide and allowed to dry. If the cells are on an agar plate, they should first be transferred to a liquid medium for dilution. The cells should form a thin, barely visible film. This can be achieved by smearing cells obtained from the surface of an agar medium or from a liquid culture. Fresh cultures must be used because as cells age, they lose their ability to retain the stain. The cells are then fixed to the slide by passing slightly above the flame of a Bunsen burner. After passing above the flame, the slide should feel warm when touched to the back of the hand, but should not be too hot. The fixed cells are then stained with the basic dye, crystal violet, for 30 to 40 seconds. The slide is then rinsed with water to remove excess stain. At this point, all cells appear purple under the microscope. Next, a solution of Graham's iodine is added and retained on the slide for about 1 minute. The iodine combines with the crystal violet to form a di-iodine complex, thereby decreasing its solubility within the cell. At this point, the cells still appear purple. The cells are then decolorized by washing with ethanol or acetone. This is the differential step. Gram-positive bacteria retain the crystal violet, whereas gram-negative bacteria do not. The ethanol or acetone should be added drop-wise, with the slide tilted at an angle until the drop coming off the edge of the slide just starts to become colorless. Even gram-positive cells can lose the crystal violet iodine complex during prolonged excessive decoloration. Excess ethanol is then washed off with water. When viewed under the microscope, gram-positive cells appear purple and gram-negative cells are colorless. Finally, the rinsed cells are covered with the counterstain safranin for 20 to 30 seconds. This stains the gram-negative bacteria pink. After rinsing with water, the slide is dried with filter paper. When viewed microscopically, the gram-positive bacteria are purple and the gram-negative bacteria are pink. Generally, the gram stain correlates with the cell wall structure among the bacteria. The ethanol is thought to shrink the thick peptidoglycan in gram-positive cells, thus retaining the dye. The thick dehydrated peptidoglycan layer of gram-positive bacteria appears to be a permeability barrier, preventing the loss of the crystal violet iodine complex. In contrast, the peptidoglycan in gram-negative bacteria is very thin and has large pores. Ethanol may extract lipids and increase the porosity, thus removing the crystal violet iodine complex.
So gram-positive cell wall. Gram-positive cell wall is made with several layers of peptidoglycan, and it has a unique proteins called teichic acids, which is only seen in gram-positive organisms, which are present on gram-positive cell wall. And the gram-positive organisms always appear as blue or purple in color, and they are more sensitive to penicillin and lysosomal action when compared to gram-negative. The gram-negative organisms are less sensitive to lysosomal action because they have one more membrane called outer membrane, which is hydrophobic in nature, which prevents the permeability. And in gram-positive cell wall, 50% of the gram-positive entire envelope consists of peptidoglycan layer. It will be 20 to 80 nanometers in thick. Gram-negative cell wall. Gram-negative cell wall consists of outer membrane, thin peptidoglycan layer, periplasmic space. There are three things. This is outer membrane, and this is thin peptidoglycan layer, and this is the inner cytoplasmic membrane. So in between you can have a space called periplasmic space here, periplasmic space. So outer membrane of gram-negative cell wall, and it is unique for gram-negative cell walls. It is only seen in gram-negative cell walls. And the bilayer outside of peptidoglycan layer intercalated with proteins. It is associates with a lot of proteins like lipopolysaccharides, porine proteins, lipoproteins, and it is the permeability barrier. It prevents the molecules to enter into the cytoplasm of the bacteria, so it prevents the lysosomal action. So lipopolysaccharides always seen here on top of the outer membrane, which prevents the permeability, and these lipopolysaccharides which are present here can also act as endotoxins. And we can also have porins here, which are made up of proteins, porin channels, and we'll have lipoproteins which always anchors this outer membrane to the peptidoglycan layer, and it also prevents the permeability. So lipid polysaccharide, that is LPS, which is very much important. The LPS can also be referred as endotoxin, and it is hydrophobic in nature. It consists of hydrophobic region called lipidae. This is the lipidae. If you see this picture, this is LPS, lipopolysaccharides, and this lipopolysaccharides has three parts. One is lipidae, which is hydrophobic in nature, and the other one is a saccharide in nature, sugar in nature, which is hydrophilic in nature. And here we can have O specific polysaccharide. The lipidae, this portion is exactly called as endotoxin, which can act as an endotoxin. These endotoxins can activate complements. They activate clotting factors, which can lead to intravascular coagulation. They activate macrophages. They increase the permeability of capillaries, which can lead to the loss of fluid from the blood circulation, which can lead to hypertension, and it leads ultimately to endotoxic shock. That means by loss of fluid from the bloodstream, we will get hypertension. Because of intravascular coagulation, there is lack of supply of nutrients to the organs, which leads to organ failure, and ultimately the patient will enter into endotoxic shock, and which is very much fatal. And there is one more part in this polysaccharide, LPS, lipid polysaccharide, that is O specific polysaccharide, this area. This O specific polysaccharide is mainly useful for adhesion, attachment, and in some bacteria it also helps in restricting phagocytosis, and it is also antigenic in nature, and we can call this O specific polysaccharide as O antigen even. Gram negative peptidoglycan layer is very thin, that is up to 10 to 20 nanomicrometers in size. And there is one more part in gram negative cell wall, that is periplasmic space. It's a gap, just a space, and in this space they consist of lot of enzymes for assembly of cell wall and membrane components, and various degradative and detoxifying enzymes are synthesized in periplasmic space. Secretory systems will be present, sensing proteins for chemotaxis, signal transduction, endoflagella can be seen inside this periplasmic space, especially for spirochetal organisms, and signal transduction. Whenever some bacteria associates with host cells, how they get the signal is through this periplasmic space. And the protein sensing chemotaxis, if at all a phagocyte is coming, how they will be moved away from phagocyte is with the help of sensing proteins, which are nothing but lipoproteins, which are present on the outer membrane and periplasmic space. And it consists of secretory system. There are a lot of enzymes that are secreted. Toxins are secreted through this periplasmic space. So the last part of cell envelope is cell membrane. The cell membrane is similar to eukaryotes, and it is a big permeability barrier. It won't allow anything to enter except the specific molecules required for the bacterium, and there is no sterols, and they can have a lot of saturated fatty acids inside this plasma cell membrane of bacteria. The main functions of cell membrane are their osmotic or permeability barrier, a location for transport systems to transport the nutrients in and out. They are the energy generating structures, because the plasma membrane of the cell membrane of bacteria is the one which acts as electron transport chain through mesosomes, involves in respiratory and photosynthetic electron transport systems, establishment of proton motive forces, ATP synthesizing through ATPase enzymes. They synthesize membrane lipids. They synthesize murine that is useful in peptidoglycan. They assembles and secretes extra cytoplasmic proteins, which are nothing but the exotoxins. Coordination of DNA replication and segregation with septum, formation of cell division. They can cleave the septum. They can helpful in DNA replication. They are helpful in chemotaxis. Wherever they find the host or cell receptors, they will move towards that area. Location of specific enzyme systems. So mesosomes are nothing but invaginations of cell membrane, and these mesosomes we can see electron transport chain where the ATP synthesizing occurs in these mesosomes, and these mesosomes are also helpful in DNA replication and the cleavage of septum, cell wall synthesis, and they are also helpful in enzyme activity. The next component inside the cytoplasm is plasmids. Plasmids are extra chromosomal DNA molecules. They are not useful for any DNA replication, but they can replicate by themselves. They are not useful for, say, bacterial functions. And there are different types of plasmid. F plasmid, which is useful for fertility. That means whenever it transmitted into the other organism, it start to replicate. R plasmid is for resistant. This R plasmid containing bacteria, most of the time the resistance to antibiotics. Col plasmid, which can produce bacteriocins. That means the bacteriocins are the proteins which can destroy the bacteria itself. That means autolysins. And Ta plasmids, which are virulent in nature. The Ta plasmid containing organism is more pathogenic when compared to normal organisms. Ribosomes. They are 70S in nature, whereas in eukaryotes it is 80S. They are smaller than eukaryotic ribosomes. 70S ribosomes in eukaryotic mitochondria and chloroplasts contain single-stranded rRNA closely related to bacterial ribosomal RNA. So that's why the bacterial RNAs are related to mitochondria. So mitochondria are evolved from prokaryotic type of cells. This is the one evidence for that these organelles are descended from prokaryotes. A DNA material, nucleoid material, and typically one large circular molecule of DNA, just circular in nature, and coiled or sometimes supercoiled, and sometimes it is associated with proteins, and you can't see mitosis or meiosis in bacteria, and the replication of bacteria is always by binary fission. Inclusion bodies. Inclusion bodies are nothing but the vacuoles which always store the nutrients required for the bacteria. They can include glycogens, inclusions in E. coli for energy source, polyphosphate or volatin granules you can observe in Corynebacterium diphtheriae, and these polyphosphates are useful for producing energy. And these are the inclusion bodies inside the bacteriums, and this is the inclusion bodies, and here you can have an inclusion body here. That's about the structure of bacteria, and we'll learn about endospores. So endospores are the dormant forms of bacteria. Normally they are formed intracellularly in unfavorable conditions. Whenever there is no favorable condition, whenever they lack nutrients, whenever the outside environment is not comfortable for the bacteria, some bacteria they will immediately changes into a resistant form or a dormant form called endospores. They start to change within the cell. They are resistant to desiccation, radiation, heat, antiseptics. So here you can see endospores. Endospores are produced as intracellular structures within the cytoplasm of bacteria, and the most important bacteria that produce endospores are Bacillus and Clostridium species. So you can observe here the endospore formation. These are bacilli, and here is the endospore formation here inside whenever there is an unfavorable condition. In Clostridium botulinum, this is the Clostridium botulinum organism in electron microscopic picture. And here you can see Bacillus organism. They're producing endospore, this white color one which is present inside. And here you can see another Bacillus organism which is producing endospores in comfortable, unfavorable condition. So endospore formation is not a mechanism of reproduction. Rather, it is a mechanism for protecting the bacteria by themselves, for survival in deleterious environment. During the process of spore formation, one vegetative cell develops into one endospore. You can see the endospore formation here. The nucleic acids are present here. They are coming together. They are coming together here, and they came and they formed a single nucleoid material here. It is forming an endospore. It forms, and you can see the small endospores here. And these are the endospores already formed. And this is a vegetative cell, and this cell start to produce the endospores. And here is the endospore coming out of from mother cell. The mother cell is called as sporangium here, and this is the endospore that is coming out. And in unfavorable condition, this endospore is resistant for many years, like Clostridium and Bacillus. They can live for years together. So classification of bacteria. So how we can classify bacteria? In a simple way, we can classify bacteria depending upon their shape, depending upon gram staining, and depending upon their respiration. Depending upon their shape, we can have different shapes of bacteria. Cocci, which are round in nature, the bacteria which are round in nature here are called as cocci. The bacillus, which are rod shaped organisms, are called as bacillus. And cocobacillus, they always look like either cocci or bacilli combined together, because the edges of this organisms are curved in nature. So in gram staining, in staining techniques, we can see this organism sometimes as cocci, sometimes as bacillus in nature, cocobacilli. And some organisms are spiral in shape like this, like corkscrew. And some organisms are diplococci, that means they are cocci organisms, and most of the time we can observe them in pairs. Pleomorphic, and we can't define the shape. They will always change their shape, so there is no specific shape for some organisms. Those organisms come under pleomorphic. You can see a Vibrio group of organisms, a curved organism like a comma shaped organisms. So depending upon shape, we'll have cocci variety, we'll have bacillus, we'll have cocobacillus, spiral in shape, diplococcus, pleomorphic in nature. Depending upon gram stain, we have two varieties of bacteria. One is gram positive, which always visible as blue under gram staining technique, and gram negative organisms, which always looks like red organisms under gram staining. So according to respiration, how we can classify bacteria says four varieties. Obligate anaerobes. So obligate anaerobic bacteria, they always requires oxygen for their metabolism. Without oxygen, they will be destroyed. So those organisms are called as obligate anaerobic. They must require oxygen. So the best example for these organisms are Mycobacterium. Second variety is facultative anaerobes, and the bacteria which require oxygen, but without oxygen also they can live for some period of time. So those organisms which can require oxygen or without oxygen, they can survive for some time. So those organisms comes under facultative anaerobes, and most of the bacteria are facultative anaerobes. Staph aureus, there are a lot of bacteria which are facultative anaerobes. Microaerophilic group of organisms. The microaerophilic organisms are anaerobic organisms, but even they have a minor concentration of oxygen surrounding them, they can resist and they can survive. So those organisms which are anaerobic but they can resist oxygen, small percentage of oxygen surrounding them, those group of organisms are called as microaerophilic organisms. So the best example for microaerophilic is Campylobacter and Helicobacter group of organism. Helicobacter lives in stomach areas, which causes gastric ulcers. And the last group is obligate anaerobes. They hate oxygen. They will be destroyed in presence of oxygen. Obligate anaerobes. These obligate anaerobes, best example is Clostridium. The Clostridium, which causes tetanus infection, which causes gas gangrene, which causes botulinum infection, is from Clostridium species. They all are obligate anaerobes. They hate oxygen. So that's why in environment they are always seen as spores, endospores.